Hello everyone. If you have already watched our videos on lectures 14 and 15 on our metabolism of proteins and amino acids, you will have seen that we came upon two new cofactors. And in this short video, sort of as part of our series on videos on cofactors, I just want to introduce you to these two cofactors that we had seen today as part of amino acid metabolism. And the first one here is pyridoxal 5-phosphate, or PLP. Right, we had seen PLP in our aminotransferase reactions, right? It's an essential cofactor in the transfer of the amino group between an alpha keto acid and an amino acid. This is actually its structure here. You don't have to memorize this structure. The business end really is this carbonyl group here, this aldehyde function. And in our case here, as part of the reaction of aminotransferases, this is a business and the carbonyl group that can capture the released amino group, can hold it in place for a while, to then hand it over to the next substrate that is coming in our aminotransferase reactions. So have a look at those reactions again in the main video if you don't quite remember them. All I want you to remember here is that we have this carbonyl group here, the aldehyde functional group, as part of this molecule that actually does the holding in place, the binding or temporary binding of the amino group. PLP is essential for us. We have to get it through our diet. It's called vitamin B6. And actually, it's not just used in aminotransferase reactions. It's actually used in a whole number of reactions. There are more than 140 PLP-dependent reactions in metabolism. For our part here in BIS103, we only focus on what PLP is doing as part of the AT reaction. As I said, it's essential. We have to get it through our diet. There actually are a number of versions that we can potentially get. One being this pyridoxal right here, another one being the pyridoxine here, and the third one is pyridoxamine here. So the difference is it's all the same structure, but we have different functional groups. Here's an amine, here we have an alcohol, and here we actually have the final pyridoxal aldehyde functional group. But if we get these other forms through our diet, we are capable of modifying them on either doing the amino transfer or the oxidation. So those are fine resources for us. And there's actually a number of foods that are rich in PLP, or versions thereof. Fish, meat, turkey are all good resources. And for the vegetarians and vegans among you, sunflower seeds and pistachios are actually quite rich in PLP. All right, so let's move on to our second cofactor that we saw today when we talked about the breakdown of glycine via serine and the general interconversion of serine and glycine with each other. So the second one here is tetrahydrofolate, or THF. It's a bit more complicated as a structure. You might have heard of folic acid, right? That's an important vitamin, vitamin B9. So much like PLP and our other B vitamins, we cannot make this compound here. We have to get it through our diet. Again, by no means you have to remember or be able to recognize the structure. You should definitely be aware that it exists, B9 as folic acid. What we can do if we get this structure through our diet, we do actually have a reductase. It's called the dihydrofolate reductase. This can actually use NADPH, so your electron carrier coming out of the pentose phosphate pathway, the PPP, and we can actually catalyze two sequential reductions here. We're reoxidizing NADP. What we're generating is now the doubly reduced form of folic acid, which is tetrahydrofolate. And so this is actually your cofactor here. Okay? This is the active form of the cofactor. What it does, it actually is a carrier of 1 carbon units, right? And it's very versatile. So it's a really important and interesting cofactor. Where you get it from, there's actually a number of foods here. Fruits and a lot of your greens are rich in tetrahydrofolate. You also will see that it's actually part of our enrichment act that requires to put certain cofactors, especially the B vitamins, into our foods. And you find it here as folate or folic acid. And it's so important that it's part of the Enrichment Act because a deficiency in folate is especially critical during pregnancy because a deficiency can actually cause birth defects before the child is born. Actually, at the very early development of a child, it can cause neural tube defects, it can cause defects in the brain and spinal cord. And these defects can set in very, very early on before a woman even may know that she is pregnant. So a continuous supply in your food of folate, especially for women, is incredibly important. And you might see right here, if you sort of look back at our cofactors, there are a number here that we now know, right? We just talked about folate. You may remember your niacin, right? That's part of your NAD. You remember thiamine here, that was part of our TPP cofactor. Riboflavin, right, was part of our FAD and FADH2 cofactors. And iron here wasn't necessarily part of our cofactors, but it's a cofactor in, for example, our heme and iron sulfur clusters, right, that were critical in the ETC. So all of these things have been deemed critical as part of supplying your food with it so that we don't have deficiencies in these cofactors. All right, so what does THF actually do? Like I said, it's a very versatile cofactor. Generally, it transfers one carbon unit and can do so at different oxidation states of carbon. And this makes it very different from biotin, right? If you remember biotin, this was our cofactor for all things decarboxylation because it's a cofactor that can transfer carbon, but it can only do so in its most oxidized form, in form of carbon dioxide. THF can do that too. It can transfer carbonyl groups, but it can do a lot more. It can actually transfer carbons at different oxidation states. So the way this works, I just show here. So if you just sort of go into the major structure, what I show here is just the business end, which is right here. And these are these two nitrogen atoms here at position 5 and position 10. So it's N5 and 10, that's what we call it. And so we can switch between different oxidation states of THF by doing a hydrolysis reaction and oxidation reduction reactions. So let's have a look at sort of starting here, right? This is what we looked at when we talked about glycine and serum metabolism. Here, THF actually switches between its unconjugated form, so both nitrogen atoms have not been conjugated with a carbon unit, and a form that actually has a methylene group here. And so this could be, for example, one way to actually get a carbon unit onto THF is by using amino acid interconversions and actually taking one carbon from the amino acid and attach it to THF. And regardless of how you got the carbon unit onto THF, you can then do a number of reversible reactions to change its oxidation state. So if you just start way at the beginning here, right, you could also, for example, use the unconjugated form. You could bring in bicarbonate from your bloodstream, and then you could use ATP hydrolysis to bring the carbon, the bicarbonate into THF, right? It will then be attached here to the nitrogen atom at position 10, and you're generating an aldehyde function here. You have your carbonyl group here. This is the most oxidized form. We call it N10 formyl THF. You don't have to remember all of these, but I want to highlight for you how these oxidation states can change. Say you have done that, right? You have brought in this most oxidized form, but you want to actually transfer a more reduced form to another substrate. What you can do is you can actually dehydrate this. So removing the oxygen function, now you're generating a methanol group. So now we have the carbon unit bound between the N5 and N10. And accordingly, this cofactor form of THF is called N5, N10 methanol THF. Now we want to reduce this even more down from a methanol. We're using NADPH again, right? From our PPP pathway. We can now reduce this further down here, right? The double bond here between carbon and nitrogen is gone. We have reduced it. This is now called methylene THF. And yet another reduction step. We're actually opening this ring. We are reducing this carbon even further. The bond will open in the process. This will now actually have been transferred over to the N5. We call this the N5 methyl THF because all we have bound now is a methyl group. And THF in these different forms now can actually transfer all these carbons in different oxidation states to another molecule. So it's a very versatile cofactor, very important one. Because it's so important, we actually use it as a target for a number of drugs. So for example, antibiotics and sulfur drugs like trimethoprim or sulfur drugs, they're actually used to inhibit the dihydrofolate reductase in bacteria. So it's fairly specific to the bacterial enzyme that can generate the active cofactor from the folate precursor. And in doing so, right, we prevent in the bacterium that it actually can generate this cofactor, and that will be lethal to the bacterium. So this is how actually these antibiotic type drugs are working. Right? We can also actually target the human dihydrofolate reductase. Sounds counterintuitive, right? We want to make this cofactor, but we can actually use it, for example, for chemotherapy. So in this particular case here, there's multiple drugs. One prominent one you would use is methotrexate. And what this actually does is that it inhibits the human dihydrofolate reductase. So we cannot make this cofactor anymore. At least it's less efficient now. And so what this targets is in the cancer cells, it inhibits histone methylation. Right? So in this case, the THF in its most reduced form, carrying a methyl group, would be used to methylate histones. And that methylation of histones is really critical for cell division. Now, because cancer cells divide much more rapidly than your normal healthy cells, the strategy here is that cancer cells are damaged by this drug much more effectively than your normal cells. So the hope is that you can kill the cancer cells by preventing this rapid cell division. So obviously this is toxic, you have to be very careful, but as all chemotherapies, essentially these are poisons. You're just hoping to attack the cancer cells and damage those more rapidly than your healthy cells and defeat the cancer that way.